Hi, I'm Owen. I'm finishing up my PhD in the Randall Lab at the University of Liverpool, and I'm going to kind of give you a filler into the prequel to Denai's work, and then also fill in some gaps in Laura's talk earlier. So thanks for MMF for having me, and thanks for the patience, because without their sort of donations, none of this is actually possible. Rather than go through in detail about what we've been hearing about for the last 3 days, the main points that I wanted to pull out of these slides was the lack of immunotherapy response that Denai has really highlighted, and then also the consensus statement placing a real key priority on preclinical models being developed for cholangiocarcinoma, and the updated 2026 consensus statement specifically mentioned precision cut liver slices. Denai's covered this a lot already, but we're looking, and Denai wrote a review looking at the immunomodulation opportunities within cholangiocarcinoma and what's already been done in the literature. But most that we could find has been done in murine models. So we wanted to sort of improve the translationability of, build a preclinical model or develop a preclinical model that actually reflects the immune tumor microenvironment in patients. When I submitted these slides, we didn't actually have any of the data. We now do have the data, so I'm just going to give you an overview of what's happening without actually showing you the data. So we looked at we took cores from patients and developed TMAs in the periphery and the center of intrahepatic, perihilar, and distal patients that were all treated in Liverpool. And we have 15 years worth of clinical outcome for these patients. And the idea of this work was to see if there was a difference between the three subtypes in immune populations and expression, if there was a difference between the core and the periphery within a patient's tumor, and then also within regions there was different expressions of exhaustion markers, activation markers, or different markers of immune populations. We saw some of the work in Laura's talk earlier. We are seeing differences between the three subtypes, and we need to explore this a little bit more. So a bit more of an overview of the slice model. So my PhD primarily has been validating this model and trying to work out the flaws. And we know that no preclinical model is perfect, but we need to understand what are the strengths and weaknesses of this model. So we can get our samples from the Royal, from the good links that we have with the surgical teams there, back to our lab within sort of 20 minutes to half an hour, and I can have them sliced in the incubator and relatively happy and stable within half an hour. So firstly, we did an overview of are the cells alive? We did a basic MTS viability assay. And the way that this works, if a cell is actively respiring, it will turn over the assay that we add and change its color. So the darker the color, the better the assay and the better the cells are and the more alive they are. We've converted this to a percentage, you can see here, just to sort of normalize between patients. And we did this in 17 cholangiocarcinoma patients, a mix of perihilars and of intrahepatics. And we didn't see a significant change over the 15 days in culture. So that was quite a good sign to begin with. We then took some of these patients' samples and put them in FFPE, which we've heard quite a lot about in the last 3 days. And we looked at the H&E staining, the CK19, CD3, and CD68 to try and look at the tumor architecture to see if that's retained over the 15 days, as well as some immune populations of T cells and macrophages. And you can see in the H&E here that they are present. The H&E here, that structurally they stay the same, and the cell populations that we're interested in are present over the 15 days. So it's all well and good looking things under a microscope and seeing if they're there, but we don't actually know what's happening at protein level. So we undertook swath proteomics, and Laura went into some detail about the differences between the two subtypes. But what I wanted to make sure, as well as looking at the differences between the two subtypes, we have 5 patients who are intrahepatic samples and 5 perihilar samples, and we looked at their protein expression over the 15 days in culture in their tissue slices. And what I was concerned about was, are these proteins that we're seeing that are differentially expressed, they're different at the beginning, are they still different at the end? Because if they weren't different towards the end, it's sort of a pointless model. If the differences that we're so interested in are collapsing into nothing, then it's not really fit for purpose. But thankfully, they did stay different. We then did some gene set enrichment analysis, which looks at known pathways, and rather than looking at the actual expression of a protein, it looks at the general shift over time and sort of adds multiple proteins together that build up to form a pathway. And what we saw here was that it was general adaptation to culture. No hallmark pathway was collapsing. So if we were seeing lots of apoptosis happening, we'd be quite concerned. But overall, it seemed quite stable in culture, especially after day 7. I did another form of clustering, which is called M-fuzz clustering, and you look at the trajectory, so how the proteins change over time, if they increase, if they decrease and rebound. And we saw similar patterns that we saw in the gene set enrichment, which is a sort of a shock response to being taken out of the body, being sliced up and then cultured, adaptation to culture, and then stabilization by day 7 onwards. So we know that there are immune cells there based on the IHC that I did, because we could see brown stains for immune cells that we looked at. We know that they're sort of stable as a model by day 7, but what I wanted to look at was then, are the immune cells that are present in these slices alive, because you can't tell that from the histology, and are they functional? And what's going on in the bulk tumour? What's different between perihylers and intrahepatics, which Laura talked about earlier? So to do this, we used mass cytometry, which to begin with has sort of similar principles to the histology that you've seen before, but then gets way more complicated after that. So you start with a protein of interest, an antibody that tags to that protein, but rather than attaching a brown stain or colour that you've seen before in previous talks, I attach a metal. And that's because if you're looking at colours, you've got a very limited range of sort of spectrum that you can actually perceive, whereas with metal, I can go off weight. So I developed a protocol where I could dissociate my tissue into a single-cell suspension, isolate the immune cells as best as I could, attach this antibody cocktail of different metals, fire it through a nebulizer and time-of-flight recorder, which then measures the weight based on how long it takes for that cell with all the different metals on to fly through the machine. And then from that, I can pick out the live cells that are single and CD45 positive. And after that, it's a bit of a sort of guess who game. You look at a single cell and you say, well, what metal's it got attached to it? And based off the metal, what protein must it have? And some cells can only express a certain combination of proteins. That's sort of the theory. And using this, I could have easily either gone through my entire 10 million cell population and manually worked out what every single one is, or you can do a sort of more time-effective method, which is using a Tisney plot, which assigns a score based on markers that I tell the computer I want to look at, so major immune lineage markers, and it then scores them and ranks them, and then begins to cluster them. So every single dot on here is a cell. I had to downsample this, or my computer didn't like it. But from what we can see from here is that we have major myeloid immune populations within our intrahepatic patients. So this is just intrahepatic, it's 5 patients. I then took their day 7 tissue slices and did the exact same process and the exact same clustering. And we can see these immune populations, there isn't a significant change over time, over these 7 days. They do shift, but we're expecting that. Also, from where I'm taking the cores from and slicing these tissue slices, it's going to be different. We know there's a lot of heterogeneity within the tumours. But it was really promising to see that there wasn't just a massive collapse in live immune cells in the tissue slices. So to sort of summarize this work, we validated the precision-cut tissue slices as a model, and I also repeated a lot of this work in normal liver, and we saw similar trends. I've had a look at the immune profiles and immune populations that we have in the tissue slices, as well as the bulk tissue, and some more work needs to be done to look at the activation status and exhaustion markers of these immune cells. That eventually can be linked to clinical data, but as this is fresh tissue that we've got, we haven't quite got that follow-up of clinical data yet. And to continue with the geomic spatial profiling to hopefully unpick a bit more what's going on in different subtypes of cholangiocarcinoma. And then leading on to Denai's work, how can we begin to manipulate this? Thanks to MMF and the patients and everyone on these slides. Thanks for listening.